Of microwave irradiation on seed germination and plant growth of pinto beans. So my main objective was just to see if there was a relationship between microwave exposure and germination rates, and then once those seeds had germinated and been planted, I wanted to see if there was a relationship between microwave exposure and plant growth. And the variables I was looking at for plant growth included the height of the plant and the total leaf surface area. So a little bit about seed germination. It's a very delicate process that relies on many external factors such as tissue composition and hydration levels. And another important thing is that the ability of the seed coat to crack may affect germination. And so I was kind of interested in seeing if heating of the seed would affect the permeability and strength of the seed exterior, and then that would have an impact on the germination rates themselves. And also, microwave irradiation is known to target water molecules, and so to see if it would have an impact on hydration levels and therefore germination rates as well. 213 pinto beans were first randomly selected and then assigned into 95 labeled petri dishes, and then these 95 petri dishes were randomly assigned into 5 groups of 19. These groups included a control, which were never exposed to the microwave irradiation, and then those that were microwaved for 5 seconds, 10 seconds, 20 seconds, and 60 seconds. The seeds, after they were microwaved, were placed on a filter paper and wet with 7 drops of water. The petri dishes were then taped closed and put in a greenhouse tray with a paper towel covering them to kind of mimic being under soil, and then they were placed under direct sunlight. After 4 days of no germination, 800 microliters of water were added to each petri dish, and they were left again, and germination was scored for every night after sunset. So once the seeds that had successfully germinated had all germinated, they were planted 30 millimeters deep in 60 millimeters of soil in Dixie cups, and these were placed directly in sunlight on the windowsill. And every morning, 10 milliliters of water was added using a graduated cylinder, and plant height was scored after 7 days just using a ruler. And the leaf surface area was scored after 7 days as well by using the Easy Leaf Area app. And this works by using the color ratios of each pixel in an image to decipher what was background image and then what was actual leaf. And a red square that's 4 centimeters cubed is placed next to the leaf, and that's used to scale and kind of calibrate the image. So this first chart shows the germination ability percentages for each group, and it was the highest for the control in those microwave for 5 seconds with 89% of seeds germinating, so 17 out of the 19. And then there was a slow decrease for 10 and 20 seconds, and those microwave for 60 seconds, they never germinated. The next chart shows the average day of germination for the groups, and it was the lowest for those exposed for 10 seconds with an average of 5.9 days, and it was the highest actually for the control with 6.7 days. And so this graph just shows that kind of general trend. And a one-way ANOVA was also run using Excel, and the results yielded a P of 0.03, so that suggests that there is a difference between treatment groups and that it may be significant. This next one shows average plant heights across each group, and you can kind of see there's a slight positive correlation between exposure time and plant height. And again, a one-way ANOVA was run, and it yielded a P of 0.497, so that also demonstrates that there may be differences between the control and the rest of the groups. And finally, this graph shows the average leaf surface area across the groups, and it was the highest for those treated for 5 seconds and the lowest for those treated for 20 seconds. And again, a one-way ANOVA was run, and it showed a P of 0.0025, which indicates that there is a pretty significant difference between the groups. Sources of error. It was hard to make sure that the same amount of soil was added to each Dixie cup, and so finding a more precise way to do that in a future study would allow for more precise results. And it was also difficult to plant all seeds at the exact same depth, and so coming up with a better way to do that may also have an impact on results. And I think in a future study it would also be cool to be able to check germination levels more frequently to give more exact data. So instead of just having the days of germination, even hours or, a different time period, to be able to have more kind of control over your variance.